Did you do the cross? The problem? Yes or no? You tried, everyone? This one. Yes. And the first thing is that I have three groups. So, and your group, you need another person. Okay. And I need the rest of you as groups. So figure out the groups, then I will give you the project next time, next day. Because based on your group, I need to find out the project. Okay, so what about this one? Have you tried? No. Okay, I'll go through this one a little bit, and because this is really important. This one. So I'll go through this one, and then probably the rest of this linkage part I will make a little bit short. That one, then I'll go for the another big topic, that is the chromosome evolution. Okay, so from here, these are the phenotypes. Okay, just think about that. These are the phenotypes. Why? Because it was test crossed. Test cross means the other gene is always recessive. If those are recessives, then these will be their phenotype. Okay. So CH, B plus N. At first, we need to find out two things. The first step is that find out the non-crossover type and double crossover type. Okay. Based on these numbers. That is the first step. Second step is that you need to find out the correct order of the genes. That is the second step. Okay. Next, you need to find out the proportions there, their ratio, and CC, that is coefficient of coincidence and interference. I will talk about that one a little bit later. But at first, the first step is that find out the non-crossover type and double crossover type. So what is the non-crossover type?
The CH,
So CH plus B plus and CN plus. These are phenotypes. Okay. And this number is 750. What is the another one? CHP CN. Number is 753. Double crossover type?
CHP CN plus.
CHP CN plus. Number 5. And
CH plus B plus CN 4.
okay. So these are main two types. Now you can just add those. These are non-crossover type. In your book you have a little bit different things, but it will be easier. This is non-crossover type NCO. This is double crossover type, total double crossover type, total non-crossover type. So what is the total of this one? Someone has calculated? This is 9. And this is 3015. I think so, right? 1503. So this is our total non-crossover type. This is our total double crossover type. Okay. Now we identified our first step. What is the non-crossover type? What is the double crossover type? Based on their numbers. Now we need to find out that which one is the middle gene. Okay. If you find out which one is the middle gene, that is fine. But you need to identify all three procedures. There are 3 genes here. You need to make everything in the middle, and I need to check whether that is true or not. So right now B is the middle gene. So take B in middle. If B in middle, that means here it is, and anything can be anywhere. Okay, I can take which one? CH is here, CH and CN. This is, and in case of non-crossover type, these are all plus, and in this side, all recessive. Right? Now, what is the double crossover after this? So here is one crossover, here is one crossover. It will result in CH plus P CN plus, and another one will be CH P plus CN. Does it match with the that one? CH B CN plus? No. So that means this one is not, but I have to show that. Okay. Now take CH in middle. If CH in middle, then it will be like here will be CH, and this could be anywhere. That this side or that side. You can take CN plus here, B plus here, that doesn't matter. Okay. This is our non-crossover type. All plus on one side and all recessive on other side. I take the double crossover here. So P plus CH CN plus. Another one will be B CH plus CN. Does it match with that one? No. Now the third one is CN in the middle. If CN in the middle, then in such case, CH plus CN P plus and CH CN plus P. Does it match? Definitely. Other two, if the other two are not, then this should be. So CH CN plus and P. CH CN plus and P. So this is the correct order of G. Okay. So now we know that this is the correct order of the gene. So CN is the middle. That means B could be either this side or that side, or CH could be either this side or that side. That doesn't matter. Now, based on that one, we need to arrange all this population. CH, CN plus, E plus, CH, CN, E. Okay, this is the correct order. Now the single crossover type. Now find the single crossover type. Single crossover between the first two genes, CN and B. So single crossover between CN and B. What is that? If there is single crossover between CN and B with the correct order, what will be the function? So this could be CH plus, CN plus, and P plus, and this one is CH, CN, and P. You need to show the single crossover between these two. Okay, so single crossover between CN and B here, right? If it is here, then the resultant will be CH plus Cn plus, and P will be here, and CH, Cn, and P plus will be here. Right? Just do by yourself. Just don't look at me. Do by yourself, match with this one. Okay? Use your brain so you have the concept clear. I know that I will do the correct one. Okay? But use your brain, do it by yourself, then match with this one. It will be easier for you to understand. Right? If this is the thing, so I need to take this one as single crossover type. So CH plus, Cn plus, P, and CH, Cn, and P plus. What are the numbers? CH plus, Cn plus, P. 102. CH plus, Cn plus, P. This is 102. And that one? 105. Total of this one? 207. Right? Okay. Now single crossover between CH and Cn. Cn is the middle. Single crossover between CH and Cn. What will be the result? I am bringing this way. So CH plus, Cn plus, and P plus, CH, Cn, and P crossover between these two.
CH plus CNP
CH plus,
40.
Cn, P,
40.
and CH, Cn plus, P plus. Right? This is this way. CH plus CNB and CH, Cn plus B plus. Right? Now, what are the numbers?
Top one is 40, bottom one 41.
40 and 41. Total is 81. Okay. What is the total number? Is 1800. Right? Total is 1800. Now, based on that one, we need to find out the total crossover between these two genes. So, single crossover between CH and Cn is how much total? 81. And double crossover percentage is how much? 9. Right? Single crossover between these two genes is 81. Double crossover is 9. So total crossover between CH and Cn is equal to this one, 90. Okay. What is the proportion? 90 out of 1800. So the proportion is 19 out of 1800. What is that? How much?
0.05.
What this is a proportion? Just probability 0.05. Okay. So total single crossover between these two, it is 207. Between Cn and P, it is 207, and double crossover is 9. So it is 216. Total. So what is the proportion? Total crossover between CN and B is 216 by 1800. 1, 2. Oh. Okay. Are you sure? Yeah. Okay. So this is our total crossover between these two genes, and this is our total crossover between these two genes. Now we need to construct the genetic map. Okay. So map is that this is the map unit. So construct the map. This is the map. First one is CH, in the middle CN, and here is B. The distance between CN and B is 0.12, or that is 12 unit. This is between these two is 12 map unit, or you can say centimorgan, 12 centimorgan. And distance between these two is 5 centimorgan or 5 map unit. This is called map unit or centimorgan. Okay? And the distance between these two genes are, that is 5 plus 12, 17 map unit. Okay? So in this way, we can construct the genetic map. Clear? Now I'll talk about the CC and intervals. Now there are a couple of things that crossover could be one, could be more than one. Anytime. It could be possible. And now just think about that. If the genes are quite apart, there is a very good chance they are having more than one crossover. This is a normal concept. But if genes are very close together, then there is a very less chance of having crossovers. Okay? So that is called the percentage. Now the percentage shows us that when it is like 12%, 12% means they are quite apart, 12 map unit. So they are having more chance of crossing overs than those two genes which are very close enough. That is the 5%. That is the correlation between recombinant percentage and the map unit. That's why we are taking the recombinant percentage as map unit. Okay. Now, if we have the crossovers can be between two homologous chromosomes and non-sister chromatids. Okay. Now the chances that crossover could be between only just two chromatids side by side. So if this is one chromosome and this is another one, so crossover could be between these two. This is one chance. Okay. Now, crossover could be between these two. This is another chance. And crossover could be between these two. Okay. If there is double crossover, then multiply all these possibilities together. Okay. If it is double crossover, then there is one chance that this crossover between these two, this is one, and crossover between these two together. That is called, the first one is two-strand crossovers. So two-strand crossovers means like just this. Double crossovers, only two strands are involved. This is a double crossover, but three strands are involved. In such case, if there is three-strand crossover, if there are only two genes, there is a chance of having the recombinant 50% detectable and 50% recombinant. If there are two genes and a single crossover, that is double crossover between only two strands, there is no such change. Why? Because if I take this one as A gene, this is B gene, this is A plus and B plus. So A here and B again, right? So there is no change at all. Though there is crossover, but there is no change at all. If there are three genes involved, there is middle gene, then you could find the differences. Okay? If there are two strands involved, in such case, we can have this A and this one is A plus. So A and here is one crossover between B and B plus. So for this crossover, we can have, in this case, we can have A, B plus. For this one, and this is straight, this is also B. So this could be A plus and B. And okay, so we can have 50% recombinant type and 50% normal. That is the wild type. And if we have four strand crossovers, then 100% is recombinant. All the four strands are involved, so there is no way that original condition retains here. Okay? Always there is a chance they will have the recombinant types. And this is four strands crossovers. But what is the importance of that one? Generally, if there are two crossovers, that doesn't matter. But we'll find later, when we'll find the chromosomal aberrations and other things, when they will pair these two chromosomes, recombinant chromosomes will pair, those two homologous chromosomes, they will form the loops. And just under microscope, if we see that kind of loops, then we can find out whether there is any crossover there or not. Okay. I think we got some kind of problem. So what is the recombinant frequencies? Sum of all single and double crossovers by total progeny. Sum of all single and double crossovers, that is the total crossover divided by total progeny. That is our recombinant frequency. And what is the interference? The thing is that the degree to which one crossover interferes with the additional crossover in the same region. So the chance is that sometimes if they are very close, then the chance is that they will not have too much crossovers. If there is one crossover, they will interfere with the other crossovers as well. Okay? So that is called the interference. And to detect the interference, we need to calculate the coefficient of coincidence. Coincidence means that all the crossovers will happen at that time. We are suspecting that if there are two crossovers, we are expecting that there will be two crossovers between these two genes. But generally, in the population, we will not find there are two crossovers. Only one crossover we can find there. So that is the interference that we expected that will be, but we are getting that one. So that is the interference between these two. Okay. So chance of having crossover, that is the coefficient of coincidence. Chance of having all the crossovers there. But when we are not getting those crossovers, that means one crossover interferes the other crossovers there. That is called the interference. Okay. So expected number of crossover is the coefficient of coincidence, and observed number of crossovers, that is the interference in a very short form. Okay. And what is interference? Interference is equal to 1 minus coefficient of coincidence. Okay. Now, in this case, in this gene map, that is our previous example here. In this gene map, we have the different distance between ST and SS is 14.6, and distance between SS and E is 12.2. In our previous problem. Okay. So, what is the coefficient of coincidence? Now, apply the rules of probability. Okay. These are two independent events. If these are two independent events, that means there could be a chance of, that is, we need to multiply those two. The probability of their crossover multiply both of them. So the probability of having crossover here, if I put it into 1, so probability of having crossover between ST and SS is 0.146. Probability of having crossover between SS and E, that is chance is 0.122. Just simply put it in 1. Okay. Now this is the total probability of having total crossovers between these three genes. Total double crossover. So what is the progeny? Total number of progeny is 755. So if we have the total crossover in 1 is this, that is the multiplication rule of probability. If we have this in 1, just multiply this one with the number, that is our total number of expected crossovers. Okay, double crossover. So we can, but we observe this one. This is our observed crossover. So total, make it a little bit more simple. Observed TCO by expected TCO is equal to the CC. Okay, observed TCO is 5 plus 3 here. Expected DCO means probability of having two crossovers. Probability of having two crossovers is 0.146. Multiplication rule 0.122. Probability of having two crossovers in terms of 1. Multiply this number with the progeny numbers. So in 755, in 1 we can get this one. So in 755 we can get this one. The multiplication, right? Simple math. And that is how much?
So,
No, that is not. 0.146 into 0.122.
Is 0.018.
0.018 into 755.
13.45.
13.45.
13.45.
This 13.45. This is the total number. Total number of expected double crossover between these three genes. Okay. Now 8 divided by 13.45.
0.594, 0.60.
Okay, I just broke into steps, different steps. Okay, so this is our coefficient of coincidence. So what we got, that means there are some interference. If we would get the whole crossovers, whole observed things, there will be no such interference. So something is interfering these things. That's why we couldn't get the expected crossover. Okay, something is doing some kind of problem. That's why we are not getting that number of expected crossovers here. If there will be no such interference here, we will get 13.45. Okay, is in case of 18, we'll get 13. So what is the interference? Interference means right now, interference is equal to 1 minus Cc, 1 minus 0.6 is equal to 0.4. Now think in other way. If the interference is 100%, interference is 1, what does it mean? If the interference is 1, what does it mean?
Does it mean that there will be no double crossover?
There will be no double crossover at all. Okay? If the interference is 0, we'll have all the expected number of double crossovers. That's why we need to do this calculation. Little bit math. Yes. Okay. Now in this case, in our problem, now can you calculate the CC and interference? So what is the probability of expected DCO? 0.5, 0.05 and 0.05 into 0.12. What is the total number of progeny? Was 1800 into 1800. What is the number?
10.8.
10.8. This is the total number. But what we got, odd TCO?
Got 9. We got 9.
9. So 9 by 10.8.
0.83.
0.83.
83%. 83%.
83%. CC. If that is our CC, that is CC is equal to 0.83, then what is the interference? Interference is equal to 1 minus 0.83.
0.17.
This is our interference. So interference is very low here. Okay, so the physical thing is that practically, actually, when we draw this kind of the process, actually it was in very earlier stage. At that time, we didn't have that concept of the multiple polygenic inheritance or other kind of things. We did the mapping of the genes. Here you can find one. This one. In Drosophila, actually, we did all this process and we have find out all these genes with their numbers, actual position by just using this technique. Using single crossover type and double crossover type, using these techniques, we could find out all these genes, the list of these genes. But right now, something is different. I'll talk about that one. So, based on that part, we did Poisson regression model. And according to Poisson regression model, what we find that actually it is called Poisson, some kind of Poisson regression model. According to that one, we have found that the recombination percentage and actual map distance is not actually perfect. It is not a straight line. Actual map distance is little bit slight difference than the recombination percentage. Okay, because of interference and other things, and not only that, because of polygenic inheritance. And using this linkage technique in human we cannot do the generation studies. There is no way, and we cannot have a lot of generations. So one thing what we can do, the pedigree analysis. Only the thing in the family that study the pedigree and try to find out whether genes are linked or not. Here in this case, we took two things. One is called the Nail-Patella syndrome. That is, Nail-Patella syndrome is capital N, this is a dominant trait, but it is rare. Okay? It is dominant trait, but it is rare. That means that we all have the recessive gene. Okay? If it is dominant trait, in case of heterozygous condition or in homozygous dominant condition, it will express in the phenotype. Okay? A group, that is A blood group, it has capital A, that is the blood group allele A, and the recessive allele N. But here is one gene containing the capital N. If there is a presence of capital N, so nail patella syndrome is expressed there with the A blood. Okay. Now in this case, the B blood group got the nail patella syndrome. A blood group didn't get the nail patella syndrome. What does it mean? Just these 3 points. Now think about that, the actual independent assortment and linkage. Can you prove that this is linked gene or not? Just these 3 genes. What do you think?
Children with type B or O have this syndrome.
Yeah, in this generation, all A blood group, all the A blood groups, they don't have that kind of syndrome. There is no syndrome at all. Only the B blood groups got, B or O blood groups got the syndrome, nail patella syndrome. What does it mean?
Means that there's like a link on the B.
If it is separated, all these 2 genes are separated, that is the separate segregation. So A and B in one gene and nail patella in other gene. So we can have all these combinations. Okay? If it is linked either this way or this way, then this chromosome will go together. In such case, just look at that one, IAN in one gene, the first person, and small I, and that is O blood group with capital N in one gene. This is the actual thing. So whenever this chromosome will go together, whenever this chromosome will go together, then the person will have the disease, otherwise not. If that is independent assortment, we can have these chromosomes in separate way. So in first generation, we could have the blood group A and nail patella syndrome. Clear? But it's a little bit complex. Just look at the chromosomal configuration there and think about that these chromosomes are going together rather than separate. If it will be separate, just at home, take a piece of paper and make them separate and do this pedigree a little bit, just these 3 things. Then it will be clear very much. Okay, so now the mapping techniques, what in our modern genetics we will use the mapping techniques. One technique is called RFLP, restriction, fragment, length, polymorphism. Restriction fragment. Do you know what is called restriction enzyme? What is called a restriction enzyme? This is a common term. Restriction endonuclease. In molecular biology, we will talk about the endonuclease and exonuclease. Restriction endonuclease. What is that? Any idea? Restricted in what? Where? Inside the nucleus. That means it is working only inside the nucleus.
Does it keep something
Yeah, you are more closer, but go a little bit deeper. Restriction endonuclease means it will cut in a specific position into the specific position. It has particular sequence there. Suppose AATCG. One enzyme is responsible for cutting that site only, not other sites. Only that specific nucleotide sequence it will cut. That is called the restriction enzymes. And previously we knew that there are a couple of restriction enzymes, but right now we know that there are hundreds of restriction enzymes are there. So this is a very good tool for us. One restriction enzyme is responsible for particularly one sequence, cut into the particular one sequence. So using the restriction enzyme, we know that end of the DNA is having that kind of sequence. Okay. So in the DNA, if we cut with the restriction enzymes, a lot of restriction enzymes, we could have some kind of fragments. So suppose this is the DNA, and these are the restriction site 1, restriction site 2, restriction site 3. There are 3 responsible enzymes here. Okay, enzyme 1 cut into this side, enzyme 2 cut into this side, enzyme 3 cuts into this side. Okay. Now in one test tube, I put the DNA, another test tube, I put the same DNA, another test tube, I put the same DNA. Okay. In one test tube, I added enzyme 1. In another test tube, I added enzyme 2. In another test tube, I added enzyme 3. Okay. Not together. In such case, in the first test tube, I will get this fragment. This is 1. This kind of fragments. Treated with enzyme 1. Treated with enzyme 2, I will have this kind of fragments. And treated with enzyme 3, I will have this kind of fragments. The rest of the thing. Okay? So this is the function of restriction enzyme, that it will cut into the specific point of the DNA. So we can have different length of DNAs there after cutting with the restriction enzymes. And did you do the DNA gel before in your lab? You did that. Other people, did you do the DNA gel? Actually, what we used to do, we used to cut the DNA sample with different kind of restriction enzymes, and as a result, we'll get this kind of fragments. Okay? So what we need to do, we need to run gel. Do you know what is gel? Gel electrophoresis?
Yeah.
Gel electrophoresis, what is that? That we have this kind of gel. These are the wells here. We can load the DNA sample here, and we will DNA runs for the positive because it is electronegative. So this is the minus point, this is the plus point. We will put the electrodes here, and the DNA fragments, those kind of fragments will separate through the gel, because the gel is a sieve-like structure, having some kind of pores there. And we'll determine the pores by ourselves. How is the percentage of gel? If it is 0.7% agarose gel, it has a little bit bigger pores. If I will make 1.25% agarose gel, it will have some small pores like that. Through the pores, the DNA fragments will separate. The lower the fragment, suppose think about that, if I push this one by ourselves, it will move a little bit. Okay? And if two of the persons will move this one, then it will go further. Okay? If I have some heavy things, I cannot move that further. But if I have the light things, it will move more further with the same pressure. Okay? So the small fragments will go here, and the larger fragments will stay here, and they will form different kind of bands. Based on the band distance, the distance of these bands are called RF. So this distance divided by this whole distance, that is called RF, restriction front or relative distance. Based on that one, we can find out the molecular. We need to run a standard gel there. We know the molecular weights there. Based on this band pattern and that band pattern, if we compare that one, we know the molecular weight of that DNA fragment. This is called restriction fragment. Okay, so the fragments produced by this way is called a restriction fragment. And using this restriction fragment and their pattern, we can find out the molecular distances. Okay, gene distance between these two. So we know the particular gene, we know the particular sequence there. We can cut that part into with the restriction enzymes. We can run that into the gel. We can find out their distances there. Based on that one, we can find out, we can map actually the genetic distance. That is called a restriction fragment length polymorphism. Okay, RFL. But it was done during 1980s. At the beginning of 90s, it was very much popular. But right now, we are always doing right now the nucleotide sequences. Another thing which is in the modern biology, genome-wide association studies. Genome-wide association studies, you can find this short term, genome-wide association studies, GWAS. Sometimes we call it as GWAS. Okay. What is this thing? Associations within population. We are not trying to find out the gene map there, but how the genes are going to the populations. What is the linkage pattern there? We need to find out. This is called GWAS. And another term is called haplotype. So what is that? Haplotype. So in nature, in normal cases, we don't have those kind of previous patterns that this gene is there and this gene is there, not something like that. We have different kind of things. Suppose here, there is C gene, but it has other genes together. They are linked. Okay. So A1, B3, C5, D4, and here is A2, B2, C4, D4, A4, B3, C5, D4. Same type of gene. They have different kind of alleles there, but the pattern is same. Okay, but they are different. They have different allelic combinations. Okay, with this group together is called one haplotype. Same kind of patterns, but different allelic combinations is called haplotype. Right now, we need to determine these haplotypes. And if a mutation arises in D group, in one of the haplotype, then if there is no crossing over, then I could find this gene in only that haplotype in the population, if there is no such crossing over. But as this is haplotype, that means they can pair together, they can have the crossovers. So the D gene, A2, B2, C4, and D, it was our first haplotype where the mutation did. Okay, always we will find the A2, B2, C4, and D if there is no crossover. So it will not come with this A2, A1, B1, C3. It will be always with A2, B2, and C4. But if I will find that gene in this combination, that it is coming with this way, that is A1, B2, B2, and C5, in this combination, this haplotype, that means there is some crossover. And in this way, in these haplotype studies, we can find out the genetic linkage different. Particularly, it is very popular in diabetic studies. Okay? And this is the modern biology, modern genetics. Okay, another two things are called somatic cell hybridization. What is somatic cell hybridization? This is another critical process. What is that? We can take the mouse cell and human cell together and we can fuse the cells. When we are fusing these two cells, that is, we are creating Frankenstein actually. Okay, the mouse cell and human cell together, we use the polyethylene glycol or PEG, and that what the function of that one, they will fuse those two cytoplasms into one cell. And when there are two things together, all the chromosomes will be together here also. This type of cells are called heterokaryon. Okay, But the thing is that eventually these two cells join together and becoming one cell. Here the chromosomes will match, but they will not form any kind of pairing during meiosis because of their different kind of pattern. Mouse cell, mouse genes are not like the human genes. Only 70% similarities are there between mouse and human genes. Okay. And another thing is that when they are doing these things, so our question is that all the chromosomes retains there or not? No. Generally, human chromosomes are getting lost from that cell. Mouse chromosomes were there. In such case, after a couple of generations, and we don't know which chromosome will be lost and which chromosome will stay there. We don't know. After a couple of generations, we'll find different types of cells having different human chromosomes. Okay? All the mouse chromosomes are there, but in these groups, suppose 4 human chromosomes still active. In this cell, suppose chromosome number 5 is active, other chromosomes are not there. In this cell, suppose chromosome number 6 and 7 is active, 1 to 5 others are not there. So we'll find different types of populations there. So what is the importance of that? What is the benefit of doing these things? Now I can identify the genes of particular that chromosomes. Otherwise, how can I do that? I cannot cut the chromosomes from the cell. I need to separate the chromosomes. This is the process of separating human chromosomes in a different patterns. Now, if I run the gel, we know the mouse pattern, all the mouse cells are there, all the mouse genes are there. If I do the restriction fragment polymorphism there, I will find their patterns in the DNA gel. Only the thing what I will not find out there, that is the exceptional cases, that is the human chromosome. Okay, so this is called somatic cell hybridization technique. And in such way, our particular gene of interest, we can identify that one. And sometimes what we are doing, we are using transgenic mice. You know what is transgenic mice? Transgenic mice is that sometimes we can manipulate the gene there. So in the ovarian cell, suppose we put some human gene into that mouse gene. Okay, now we develop the mouse. And so that mouse will contain one human gene or couple of human genes with their own gene. So we can treat those mouse with the same human drug so they can affect that. I had one kind of breast cancer mouse gene where the cancer was developed after 9 months automatically. The breast cancer gene was there in the mouse, but that was a human gene. And sometimes you can hear the term nude mice. Nude mice means they have no kind of immunopotency there. Okay, and they are very expensive, almost like $1,200 per mice, and you have to maintain a very good chamber because any kind of infection can kill the mice. Okay, and they are actually what we are doing. We are taking out. They are all these antibody production genes. We are taking out all these genes, and so they are not immunocompetent anymore. Another technique was that deletion mapping. So what is deletion mapping? Suppose we need to find out our gene of interest. We can have, yes.
I was going to say that children, they currently have immunodeficient mice or whatever, and they're giving the leukemia cancer that the kids have to the mice, and they actually cured a couple of kids for the mice. And even when they have tumors, they're generally technically euthanize them right after that. And but if the leukemia is being treated correctly, then you have to. But they'll get like random tumors from their mouse cells. But even though we were treating and fixing the human cells.
Yeah.
So
that is the transgenic mice. I think they are using the transgenic one.
yeah, we have certain immunodeficiency rooms where you're secluded to it.
Actually, the nude mice, particularly the immunodeficiency one, nude mice is sometimes what do we need to do?
Well, I guess I just think we radiate them completely and then give them.
That is another treatment possible because when we talk about the cancer, we'll know about that because the radiation is one procedure for treating cancer. Cancer is cannot be cured anyway, but cancer can be treated, so we can keep that thing at that condition. It will not progress anymore. That is the current treatment is something like that. We don't have any medicine for curing cancer. So in such case, leukemia is the cancer, that is the blood cancer, and we can keep that as is in that condition. And there is one thing, is a very sad thing. One 10 years old guy, old boy, actually he was from Mississippi, I think so. He had leukemia, and he told that to the doctor, I will not take any kind of chemotherapy. Why? Because all the chemotherapy drugs are poisons, and that is true. The lawyer, the doctors, nobody could tell him that you need to take these drugs because he was legally true. Chemotherapy drugs are killing the cells, not only killing the cancer cells, killing the some regular cells also. And that boy didn't take any kind of chemotherapy and died just after 2 months. Okay, and legally nobody could allow him to take this drug. So we really don't have any kind of things. So that radiation and other things, you need to develop the cancer there, you need to develop the leukemia, just put that is the treatment pattern. And in every type of cancer, we are doing these kind of things, either chemotherapy and the radiation. Radiation is the first thing. Yeah. Again, this is kind of deletion thing. Well, what is happening here? Suppose we have a gene of interest, and if we can delete that gene from one chromosome, or that gene is deleted from one chromosome, then any case that gene will not have their allele on the opposite chromosome. It will come into the population all the times. If I don't have another chromosome, the homologous chromosomes are something like this. So if it is A plus and A, then they will produce the heterozygotes and other things. But if I will not have this gene here, in any case, the A will come to the phenotype, that character. So this is the way that if that gene is outside the deleted part, then it will express all the times. If it is within the deleted region, or in that area or the other part of the chromosome, then it will not express. So in such way, sometimes we can map the genes. But this is the new technique, which is called the fluorescence in situ hybridization or FISH technique. What is happening here? So all those techniques are older techniques. This is the modern technique right now. Not modern, it is almost like 25 years we are using these techniques. So what is happening here? Suppose this is our gene of interest. Now we know the DNA sequence. So we know the sequence of DNA. Okay? We can identify the sequence of the gene, particular gene. Now we need to find out think about that, when we are taking the DNA from a cell, we don't know which DNA is from which. There is no way. We can take the whole cell, we can extract the DNA, all the chromosomes will be broken, and the DNA, all the total DNA will be under the tube. So we don't know which DNA is from which chromosome. There is no way. So we can find out the gene, identify the gene, we can do the sequencing, we can do the proteomics, we can find out this is the gene, but we need to find out what is the position of the gene in which chromosome. That is our main question. How can I do that? The thing is that you can make some probe according to that sequence. Probe means another small segment of DNA, okay, which is just complementary to that sequence. When we will do the replication, then we will understand what is the complementary sequence means. That means that DNA will match with that DNA in the chromosome. Our probe will match with that DNA in the chromosome. Now we can do these things by ourselves because making the probe is very cheap right now. We can have this kind of primers just like 10 cents, like that. In $1, we can have a lot of primers. We can design it by our labs. There is no way. just take the nucleotides and add it in a chemical process, and you can do that kind of forward primer, reverse primer probes and anything. With that probe, add a fluorescence dye chemically in the lab, produce that one according to that DNA sequence. Now inject that probe into the cell, and cell is dividing all the time. When there is a division, there is a replication, DNA replication. During the replication, your probe will be attached to the original DNA. You have the fluorescence dye attached to your probe. Now take this cell, make the karyotype, arrest the metaphase. Karyotype means you need to arrest the metaphase and you need to observe the cell. Then you can find this fluorescence there. From this fluorescence, you can find out in which gene it is. So this is called FISH technique, fluorescence in situ hybridization technique. Hybridization means you are using one DNA probe to hybridize with that DNA. That is called the hybridization. In situ means in the cell, and fluorescent, you are using the fluorescence tag. And as in a short form, this is called FISH technique. And this is very, very common right now. And we know that there are a couple of things here, that is the recombination rate. So why can't we use the recombination technique right now? One problem is that for the human that we cannot have that much generation. Another thing is that they can vary among different conditions. They can vary among species. So the recombination in human is higher than the recombination in mice. Okay. So with the same gene, we cannot do that. We have a couple of common genes. In the ape, we are carrying the ape genes in our body. Okay, but that is not active. But we cannot find out that there is recombination also, but their recombination frequencies are different. And among chromosomes of a single species, the chromosomes 21 and 22 in human are having the highest rates than chromosome 2 and 4. Two chromosomes in the same cell, but these two chromosomes 21 and 22, they have high rate of recombination, high rate of crossing over, but these two chromosomes, they are not having high rate of crossing over. Even in case of between sexes, the human autosomal chromosome or females undergo 50% more recombination, 50% more recombination than males. In males, the crossing over frequency is less. In female, the crossing over frequency is high. So in such case, it is very, very difficult to find out the gene location just starting the progeny. Okay. But we have some kind of recombination hotspots. Hotspots means certain area of the chromosome where the recombination rate is very high. We don't know why. We don't know why, but there the recombination rate is very high. Even within two genes, there are 4 or 5 recombinations there, 4 or 5 crossing overs. This is called the recombination hotspots. And these are everything. And generally, the recombination hotspot is very low near centromere. These are some kind of things you need to know. I think this is all about the linkage. So for the linkage, the main part is that what is linkage? What are the linked genes? This is the main thing we need to think about. Gene mapping, how to do the gene mapping. And probably that will not be in your question, but probably I will give you some kind of multiple choice based on this gene mapping. The position of the chromosome, or which one is the double crossover, which one is the non-crossover. If you will have single crossover, what will be the progeny like this? Not more than that. And what is haplotype? Haplotype is really very, very important. Okay. Now I'll start the next one. Next one is very, very important. And I didn't put this slide yet there, but I will put it, I will modify the slide a little bit because I didn't find this slide very good. My older one was really, really good. I'll combine these two slides and I will give the slides there in the next day. Next day, today, give me the total, that is, I have sum of 3 groups. I need 2 more groups and give me the group members. I will create the pages, and next day I will do the lottery for your project. Something like gene therapy, something like BRCA gene mutation. How is that? Is it good? One thing is that I need to tell you everybody that your project doesn't mean that you need to get the full points there. Not only that, but also is that we need to learn the subject. So what you are doing, you need to tell other people about that total subject. So your project should be like that. It should be informative. Okay. Generally, you will get good marks. That doesn't matter. But the thing is that we need to learn these things. Even I need to learn from it. Okay. Penetration of the genes, particularly if you get the project of BRCA gene, what is the penetration of BRCA gene into the population? You need to show these things, and this is mainly based on the genetics. You know, the metacentric, some metacentric different parts of chromosome. We already discussed that thing. But what is karyotyping? We talked about that one. Karyotyping. Karyotyping means to arrange the chromosome according to their length. You need to do that. You need to put the homologous chromosomes together. How can you identify the homologous chromosomes? That is the main thing. If the four chromosomes are of around same height, how can you identify the homologous chromosomes? Okay. Then for that one, to identify the homologous chromosomes, we have certain kind of banding technique. So the chromosomes are like this. Based on this banding pattern, we can match the chromosomes. One banding pattern, this is a very common banding pattern, is called the G bands or GM sustain. Generally, CG region, they can be stained, and other parts they are not stained. So we can identify these parts are CG rich and these parts are AT rich. Q bands using the quinacrine mustard. These quinacrine mustard GMs they are all called stains. So what is stain? Do you have any idea? Stain using biology. What is stain? Gram stain is for bacteria. You cannot visualize everything there clearly. Okay. We have some kind of bright field microscopy or phase contrast microscopy to see the three-dimensional structure of the cell, three-dimensional structure of the chromosome. Electron microscopy, that is very, very expensive, but we can find out the chromosome because in electron microscopy, we don't have any kind of stains. Okay, it is just black and white. But the stains are like that. There are specific chemicals stain the specific part of the cellular organelles. Okay. Suppose this GM sustains quinacrine mustard and those kind of things, they actually stain the chromosomal parts. That is the DNA-rich segments. But we have some kind of other stains which can stain the actin myosin fibers in the cell. We can stain only the mitochondria. Which can stain only the Golgi bodies like that. But we need to visualize all these things. That's why we need to use the stains. And based on this one, we have, this is not only four kind, we have another kind of that is called the NOR banding pattern. NOR means the nucleotide organizer region. So we nucleolus organizer region. What is nucleolus? We found it generally in the interface state of nucleus, use that nucleolus. So we can do the banding to find out those kind of things. This is the banding pattern. Different kind of bands are there. We need to match those bands to find out the homologous pairs. This is probably GM sustained slides. This is another banding pattern. Now, the thing is that chromosomes are going from one cell to another cell as a set. Okay. But sometimes, if something will happen during anaphase, during metaphase, if the spindle fiber break or other things, they will create some kind of different rearrangements. Okay. These rearrangements are called the chromosomal aberrations. I need to change these two slides quite often. So the chromosomal aberrations Any difference, any disturbances in their chromosome structure or chromosome number is called chromosomal aberration. That is called the chromosome variation. Okay. There are mainly of two types. One is called the structural aberration, and another is called numerical aberration. Structural aberration means any problem or any disturbances in their structure. That is called the structural aberration. Any alteration into their number, that is called the numerical aberration. We'll talk about the numerical aberration later. At first, think about the structural aberration. There are four types of structural aberrations. One is called deletion, duplication, inversion, and translocation. Okay. Deletion means deletion of chromosome part. Duplication means one gene twice in number, or a segment of chromosome twice in number. That is called duplication. Inversion means that gene is in other word inversion. And translocation means one part, a segment of one part, one chromosome is going to the another chromosome. Now this translocation, there are similar kind of things, breaking of a chromosomal segment, going to another chromosome. Previously we saw these things, breaking of one chromatid and changing their into the sister chromatid. That pattern, that is called sister chromatid exchange. Breaking of chromatid from non-sister chromatid of homologous chromosome to the other part, or non-sister chromatid of another homologous chromosome, that is called crossing over. Now, breaking of chromatid from one chromosome, supposing from chromosome number 1, and it is going to chromosome number 2, that is the non-homologous chromosome, that is called translocation. There are three things, but the events are different. Translocation can be a two type. One is called reciprocal translocation and the Robertsonian translocation. And this Robertsonian translocation actually creates the Down syndrome. That is another thing. So there are four things that is called the rearrangement. When there is rearrangement, rearrange starts with DNA breakage. So any kind of DNA breakage. And when can we find the DNA breakage? You need to have a little, do you have a little bit idea about DNA replication? Any idea about DNA replication? During DNA replication, what is happening that the DNA is uncoiled. The DNA is a coiled structure. It becomes uncoiled. So suppose if you will take some kind of thread, what is the coiled thread, which we can find in the stores. If you want to uncoil that one, those threads, those who are coiled like this way, if you need to uncoil that one, then which part you are uncoiling, that is becoming uncoiled, but the other part having some kind of tension. Okay, it is becoming more thicker, more dense like that. In DNA, it is happening also because DNA is coiled. When you are trying to uncoil one part, another part is having that kind of tension. One enzyme is there called topoisomerase. What is the function of topoisomerase? They will cut the strand there and just release the tension and join that part. Okay, this is the function of topoisomerase, just relieving that kind of tension by cutting the DNA strands. In such case, DNA can be broken in one part anytime. During that broken part, there is a chance of mismatch of the DNA. Okay, so DNA starts with the breakage, and generally, if there are double strand breaks, they are generally lethal. If one gene is missing, generally it is creating some kind of lethal activity. Repair system in the cell correct the double strand breaks, but joining back them. Repair system means that is the function of the topoisomerase. And there are some mutation also. During mutation, it can happen. So if two ends of the same break are joined, then DNA structure is restored. If not, then it will make chromosomal rearrangement. So if this is DNA, two strands are broken here and they can join to each other, then it is fine. If they cannot join to each other, they can join in this way. That is called the crossing over. If it is single, that is homologous chromosome. If they are two different chromosomes, this is chromosome 1 and this is chromosome 2, these parts broken, and this can join to this one, this can join to this one, then there is rearrangement. Even it can happen that, suppose, if a chromosome there is A, B, C, three genes are there, this is broken in some way, and then during rejoining, it can comes to this, just opposite. Okay, so this is the rearrangement. And when there are two things, I'll talk about this one a little bit later, that if it involves the centromeric part, suppose this is a centromere here, and we have three genes here. This part is broken. Here is the broken part. It is joining to another chromosome, even the same homologous chromosome. This part is cutting out and joining here again. So in such case, what will happen? This C gene having no centromere at all. Okay, but this chromosome having two centromeres. It can happen. If there is no centromere at all in the chromosome, it is called eccentric chromosome. And in case of eccentric chromosome, if there is no centromere, there is no kinetochore. If there is no kinetochore, it cannot attach to the spindle fibers. It cannot go to the cell division anymore, and the lysosome will destruct that chromosomal segment. So it will be lost. That gene will be lost. In case of dicentric chromosome, it is having the same problem. Same problem means if two spindles, one spindle is attaching here, another spindle is attaching here, what will happen? Where will it go? Either it need to break up in something, or there will be no cell division at all. This is called dicentric chromosome, and if there is dicentric chromosome, it will form a bridge-like structure during anaphase, and that is called anaphase bridge. Okay, just this part, deletion. Deletion means just something, one gene is deleted. Here, the C and D, if we have a break here and we have a break here, this CD part, if it is deleted from that original chromosome, and this part, this E gene rejoins with A and B, then in this chromosome becomes shortened, losing C and D genes, and this is called the deletion. And according to that one, the deletion can be with the centromeric region, can be outside the centromeric region. Deletion can be intragenic within a gene, only a single gene, or can be deletion of several adjacent genes like this one. This is called deletion. Okay, I think I'll stop. There are a lot of things to study. For the deletion, we need to build the pairing, then we'll find the loops there. And this is very, very important, particularly for this chromosomal aberration class. And there are a lot of genetic syndromes are associated with this structural aberration and numerical aberration. Couple of problems that the projects will be based on this. I'll give one structural aberration project, one numerical aberration project, and couple of molecular biology. This is the gene therapy. Who is interested in gene therapy? Okay, I'll do the lottery.
I was here. I wasn't here on Thursday.
So do you have the group?
No, I'm not even sure.
Just call these other people and fill out this group. We already have this group. And who are interested in the group?
I don't know anybody's names.
I'll be in the group with you on questions.
I'm Batty.
I don't know how many you have.
I think you're on each group.
I don't know.
Three people it looks like. I think it's three, I guess.
Usually just want three, is that the length?
Yeah,